Hello there, and welcome to the Deep Sleep Science channel. I'm so glad you've joined me tonight. Tonight, we descend into the quiet battlefield within you, beneath your skin, flowing through rivers of blood and lymph. An invisible universe unfolds. Billions upon billions of microscopic guardians patrol your body every moment, silently protecting, repairing, and remembering. This hidden world operates in near total silence, a slow and ancient dance that has been perfected over hundreds of millions of years. Your immune system is more than just a defense. It is a memory keeper, a sculptor of your body, a library of every battle ever fought. It recognizes self from stranger with astonishing precision. It sends chemical whispers across vast cellular distances. It remembers infections from decades ago and patrols for dangers that have not yet arrived. Most remarkably, this entire symphony of protection happens without conscious thought, without your awareness, like the quiet turning of the earth beneath your feet. Science is only beginning to understand the elegance of this hidden guardian, and what we've discovered is more beautiful and mysterious than we ever imagined. If you enjoy these gentle journeys, consider liking and subscribing, or share your thoughts in the comments. But for now, there's nothing you need to do. Just let yourself settle. Let your breath slow, and let the quiet vastness of your inner universe open like a calm horizon. Now, if you're ready, let's begin. Your immune system remembers every infection you've ever had, storing the memory for decades in specialized cells. Within the hidden chambers of your lymph nodes and bone marrow, memory cells stand as silent sentinels, preserving molecular portraits of every pathogen you have ever encountered. These cells can remain dormant for years, sometimes for your entire lifetime, waiting patiently in the darkness. When a familiar invader returns, these ancient guardians awaken instantly, mounting a response so swift and precise that the infection is often defeated before you even notice symptoms. This cellular memory is why many childhood diseases touch us only once, why our bodies become wiser with time, why we carry within us an invisible archive of our own biological history. It is a form of immortality written not in books, but in the language of proteins and receptors, a library that grows richer with each passing year. A single drop of your blood contains up to 25,000 white blood cells, all silently patrolling for threats. Imagine holding a single drop of water between your fingertips, something so small it might evaporate in moments, and yet within that tiny sphere, an entire army marches. These white blood cells drift through the crimson current like ships on a vast ocean, scanning the biochemical horizon for signs of danger. Some are hunters, seeking out bacteria and viruses with predatory focus, while others are messengers, carrying warnings from distant tissues. And still others are memory keepers, holding the wisdom of battles fought long ago. They move without rest, without pause, flowing through vessels narrower than a human hair, squeezing through spaces that seem impossibly small, all in service of your protection. It is a reminder that you are never truly alone, that even in your smallest parts, a tireless vigilance continues. 5 liters of blood and lymph flow through your body, carrying immune cells to every corner of your being. This is nearly the volume of a large pitcher, a river that loops endlessly through the landscape of your flesh, reaching from the crown of your head to the tips of your toes and back again in less than a minute. The blood moves in pulsing waves, driven by the steady drumbeat of your heart, while the lymph drifts more slowly, propelled by the gentle compression of your muscles and the rise and fall of your breath. Together, they form a circulatory web of staggering complexity, a transportation network that delivers immune warriors to inflamed tissues, removes the debris of cellular battle, and carries chemical messages across vast biological distances. Every organ, every tissue, every cell is bathed in this protective tide, a ceaseless flow that sustains life itself. In this way, your body is less a solid thing and more a flowing river, constantly renewing itself, constantly in motion. Your bone marrow produces over 100 billion new white blood cells every single day, deep within the hollow spaces of your bones, in the soft red tissue that fills your skeleton like hidden treasure. A factory of staggering scale operates without pause. Here, stem cells divide and differentiate, transforming into the diverse army of immune defenders that will patrol your body. 100 billion cells, a number so vast it defies easy comprehension. More than 10 times the number of stars in our galaxy, born fresh each day within the quiet darkness of your marrow. Some will live for hours, others for days, and a precious few will survive for decades, but all begin their journey in these silent chambers of creation. It is a production line that has run without interruption since before you were born, that will continue until your final breath, a testament to the body's relentless commitment to its own survival. Lymph is a clear, colorless river that flows parallel to your blood, carrying immune soldiers throughout your tissues. Unlike blood, which rushes through arteries with the force of your heartbeat, lymph moves with a different grace, drifting slowly through its own network of delicate vessels, pushed gently forward by the contraction of your muscles and the expansion of your lungs. It is a ghost river, invisible to the eye, carrying away the fluid that seeps from blood vessels into the spaces between your cells. Within this pale current float immune cells on constant patrol, drifting from tissue to lymph node and back again, screening every drop for signs of infection or damage. The lymph carries the waste of cellular life, broken proteins, dead cells, fragments of bacteria, delivering them all to filtration stations where they can be processed and removed. It is a quieter circulation, a slower rhythm, but no less vital than the crimson tide of blood that flows beside it. Your thymus gland, hidden behind your sternum, is where T cells go to school and learn to recognize danger. Nestled in the upper chest, just behind the breastbone, this small gland serves as an academy for the immune system's most sophisticated warriors. Immature T cells arrive here as blank slates, carrying the potential for countless different specificities, and within the thymus, they undergo a rigorous education. They are tested against thousands of self-proteins, and any cell that reacts too strongly to the body's own tissues is eliminated, culled before it can cause harm. This is how the immune system learns the most fundamental lesson of all: the distinction between self and other, between what must be protected and what must be destroyed. The thymus is largest in childhood, when this education is most urgent, and gradually shrinks with age. But even in adulthood, it continues its quiet work of training, ensuring that only the wisest and most discriminating cells are allowed to patrol your body. The immune system can distinguish between trillions of different foreign molecules with remarkable precision. This is perhaps the most astonishing feat of all, the ability to recognize an enemy never before encountered, to detect the chemical signature of a virus that has just emerged halfway across the world, to identify a bacterium that no human immune system has faced in generations. The secret lies in diversity. Your immune system generates receptors through random genetic recombination, shuffling segments of DNA like cards in an infinite deck, creating billions upon billions of unique recognition molecules. Somewhere among this vast repertoire, there exists a cell capable of binding to almost any foreign structure imaginable. When that rare specific cell finally encounters its perfect match, it multiplies rapidly, cloning itself thousands of times, transforming from a single sentinel into an army tailored precisely to the threat at hand. It is pattern recognition elevated to an art form, a biological miracle that unfolds in the darkness of your tissues every moment of every day. Natural killer cells patrol your body, searching for cancerous cells before they can grow. These warriors bear a fitting name, for they do not wait for instruction or activation. They are born ready to kill. Natural killer cells drift through your bloodstream and tissues, constantly scanning the cells they encounter, searching for the telltale signs of transformation or infection. They recognize what is missing rather than what is present. Healthy cells display identification molecules on their surface, a molecular passport that says, "I belong here." But cells that have become cancerous or infected with certain viruses often lose these markers. When a natural killer cell encounters a cell without proper identification, it moves in swiftly, releasing enzymes that trigger the target cell's self-destruction. This silent culling happens constantly, a quiet pruning of damaged cells before they can multiply into something more dangerous. You are protected not only from invaders that enter from outside, but from the small rebellions that arise within, cells that forget their purpose and begin to grow without limit. Your spleen filters your blood, removing old cells and capturing pathogens in its quiet embrace. Tucked beneath your left rib cage, this fist-sized organ receives nearly a quarter of your cardiac output with every heartbeat, making it one of the most blood-rich tissues in your body. As blood flows through the spleen's intricate network of channels and chambers, specialized cells examine every passing red blood cell, every platelet, every white blood cell, assessing their condition and function. Aged or damaged cells are pulled from circulation and gently dismantled, their components recycled for use elsewhere in the body. Meanwhile, immune cells stationed in the spleen screen for pathogens, particularly bacteria that travel freely in the blood. The spleen also serves as a reservoir, holding a reserve supply of blood and immune cells that can be released during emergencies, when demand suddenly exceeds supply. It is both a filter and a fortress, a checkpoint and a barracks, working in silence to maintain the purity and readiness of your blood. Antibodies are Y-shaped proteins that lock onto invaders like keys fitting into ancient locks. Each antibody is a marvel of molecular architecture. 2 long chains and 2 short chains, folded into a precise 3-dimensional structure, with binding sites at the tips of the Y that can grip specific molecular shapes with extraordinary specificity. When an antibody encounters its target, a protein on the surface of a virus perhaps, or a toxin produced by bacteria, it binds tightly, marking the invader for destruction or directly neutralizing its harmful effects. These proteins float through your blood and lymph by the billions, each one a tiny key searching for its specific lock. Some antibodies can cross the placenta, passing from mother to developing child, while others are secreted into mucus and tears, coating the surfaces where invaders are most likely to arrive. They are the immune system's long-range missiles, able to tag and track enemies throughout the fluid spaces of your body, turning invisible threats into visible targets. Your gut contains more immune cells than any other part of your body, a hidden fortress in your belly. The lining of your intestines represents the largest surface area where your body meets the outside world. If it were unfolded and spread flat, it would cover an area roughly the size of a tennis court. Along this vast frontier, more immune cells stand guard than in all your lymph nodes, spleen, and circulating blood combined. They face an impossible challenge: to tolerate the trillions of beneficial bacteria that help you digest food and synthesize vitamins, while remaining vigilant against the dangerous pathogens that might slip in with every meal. This requires a delicate wisdom, a nuanced understanding that not all foreigners are enemies, that some strangers are essential to survival. The gut immune system learns this discrimination gradually, trained by exposure to food and microbes from birth onward, developing a tolerance that is both sophisticated and essential. It is in the belly, then, that your immune system achieves its greatest feat of diplomacy, maintaining peace with a vast foreign population, while never lowering its guard. Lymph nodes swell when fighting infection, because they're filling with immune cells rushing to battle. You may have felt them yourself, small, tender lumps beneath the jaw when fighting a throat infection, or in the armpit or groin during other illnesses. This swelling is not a malfunction, but a sign of mobilization, evidence that your immune system has detected a threat and is mounting a coordinated response. Within the affected lymph node, millions of lymphocytes are dividing rapidly, cloning themselves to create an army tailored to the specific invader. The node becomes a staging ground, a command center where immune cells gather, receive instructions, and prepare for deployment. Blood flow to the area increases, delivering nutrients and oxygen to fuel this cellular multiplication, while the lymph vessels drain the battlefield, carrying captured pathogens and cellular debris back to the node for analysis. The swelling gradually subsides once the infection is cleared, but the node retains memory cells from the encounter, standing ready should the same invader ever return. These small bumps beneath your skin are monuments to battles won, temporary fortifications that dissolve back into silence once victory is secured. Your skin is your body's largest immune organ, a vast protective barrier covering nearly 2 square meters. Every inch of your skin is a fortification, a layered defense that keeps the microbial world at bay. The outermost layer consists of dead cells, flattened and tightly packed, forming a physical barrier as impenetrable as brick and mortar. Beneath this lie layers of living tissue, patrolled by specialized immune cells that sample the environment constantly, alert for any breach. Your skin secretes oils and acids that make its surface inhospitable to many bacteria, while resident immune cells stand ready to sound the alarm at the first sign of invasion through a cut or wound. The skin is also a sensory network, densely packed with nerve endings that can detect pain, temperature, and pressure, providing early warning of potential damage. Even the bacteria that live naturally on your skin, the microbiome that coats you from head to toe, serve a protective function, outcompeting dangerous species for space and resources. Your skin is not merely a covering, but a living interface, the boundary where you end and the world begins, constantly negotiating the terms of that border. White blood cells can squeeze through the walls of blood vessels to reach infected tissues. This process, called extravasation, is a feat of cellular acrobatics that seems almost impossible. Blood vessels are lined with tightly joined cells, forming a sealed tube through which blood flows. And yet white blood cells can detect chemical signals from inflamed tissue beyond the vessel wall and respond by pushing their way through. They first slow down and begin to roll along the vessel's inner surface, sensing their environment through specialized receptors. And when they detect the right combination of signals, they flatten themselves and slip between the endothelial cells, squeezing through gaps that seem impossibly small. It is like watching a person slide between prison bars, changing shape, to fit through a space that should not accommodate them. And yet, white blood cells accomplish this migration thousands of times per second throughout your body. Once through, they follow the chemical gradient toward the source of infection, crawling through tissue like microscopic explorers navigating a dense jungle. The ability to leave the bloodstream and enter tissues is what allows immune cells to reach infections wherever they occur, to bring the battle directly to the enemy rather than waiting for the enemy to come to them. Your immune system produces fever deliberately, creating an environment where many pathogens cannot survive. When you feel the rising warmth of fever, the aching muscles, and the strange clarity that comes with elevated temperature, you are experiencing a weapon wielded by your own body. Fever is not an accidental byproduct of infection, but a carefully calibrated response, triggered by chemical signals called pyrogens that reset the thermostat in your hypothalamus. The higher temperature serves multiple purposes. Many bacteria and viruses replicate more slowly in the heat, giving your immune system time to mount a response, while the fever simultaneously accelerates the activity of immune cells themselves, allowing them to move faster, multiply more quickly, and work with greater efficiency. Iron, which many bacteria need to grow, becomes less available at higher temperatures, further hampering the invaders. For most fevers, the discomfort you feel is a sign that your body is winning, that it has taken control of the battlefield and shifted conditions in its favor. The fever will break when the threat has passed, the thermostat resetting once more to its normal range, leaving you exhausted but victorious. Memory B cells can live for your entire lifetime, remembering infections from childhood. Somewhere within your body right now, drifting through lymph nodes or nestled in the quiet spaces of your bone marrow, are cells that remember the measles virus from when you were 5 years old, the chickenpox from when you were 7, every significant infection you have survived. These memory B cells are the keepers of immunological history, each one trained to recognize a specific pathogen and capable of springing into action if that pathogen ever returns. They divide very slowly, if at all, existing in a state of patient waiting, conserving their energy across decades. When their specific enemy reappears, they awaken and transform, multiplying rapidly and maturing into antibody-producing plasma cells within days. This is why second encounters with the same disease are often mild or even unnoticeable. Your immune system does not need to learn the threat from scratch, but can draw upon its deep archive of experience. In this way, you carry your history within you, at the cellular level, a living memory that shapes your present and guards your future. Tears, saliva, and mucus all contain antimicrobial enzymes that quietly destroy bacteria. Every surface of your body that meets the outside world is coated in protective fluids, and each of these fluids is more than simple water. They are chemical warfare agents, mild enough not to harm your own tissues, but devastating to many microbes. Lysozyme, an enzyme found in tears, can break down the cell walls of bacteria, causing them to burst and die within minutes of contact. Saliva contains not only lysozyme but also lactoferrin, which binds to iron and starves bacteria of this essential nutrient, and immunoglobulin A, an antibody that can neutralize pathogens before they establish infection. The mucus that lines your respiratory and digestive tracts is a sticky trap, capturing microbes and sweeping them away through the coordinated beating of tiny hair-like cilia, while also containing enzymes and antibodies that attack anything caught within it. These defenses work continuously and invisibly, destroying countless potential infections before they can even begin, so that most of the bacteria and viruses you encounter each day never make it past these first gentle barriers. Your immune system replaces itself approximately every 7 to 10 days, constantly renewing its army. The white blood cells that patrol your body today are not the same ones that protected you last week or will guard you next week. Most immune cells have relatively short lifespans, living for days or weeks rather than years, and must be constantly replaced by new cells emerging from your bone marrow and thymus. This rapid turnover ensures that your immune system remains fresh and responsive, that damaged or exhausted cells are removed before they can fail in their duties. It is like a river that appears constant, but is actually composed of ever-changing water, or a flame that burns steadily while consuming and replacing its fuel. The exceptions are the memory cells, which can persist for decades, providing continuity and wisdom to this otherwise transient population. This combination of rapid renewal and long-term memory gives the immune system both flexibility and stability, the ability to respond to novel threats while never forgetting the lessons of the past. Macrophages are large immune cells that engulf and digest bacteria, viruses, and cellular debris. Their name means big eaters, and it is perfectly fitting. These cells are the custodians and scavengers of your body, constantly cleaning up the microscopic messes that accumulate in your tissues. When a macrophage encounters a bacterium or a fragment of dead cell, it extends arm-like projections that wrap around the target, pulling it inside where it is enclosed in a membrane-bound compartment called a phagosome. The macrophage then floods this internal prison with enzymes and reactive chemicals that break down the captured material into its constituent molecules, which can then be recycled or expelled. Macrophages also play a crucial role in inflammation and healing, secreting chemical signals that attract other immune cells and coordinate the repair of damaged tissue. They are found in virtually every tissue of the body, from the lungs where they clear inhaled particles, to the brain where they prune unnecessary neural connections, to the liver where they filter the blood. Silent and tireless, they maintain order in the cellular world. The complement system consists of over 30 proteins that work together like a cascading waterfall of protection. These proteins circulate in your blood in an inactive state, waiting patiently for the signal to spring into action. And when that signal comes, often the binding of an antibody to a pathogen, they activate in sequence, each protein triggering the next in a carefully choreographed cascade. Some complement proteins coat the surface of invaders, marking them for destruction by phagocytic cells, while others assemble into attack complexes that punch holes through bacterial membranes, causing the bacteria to burst from osmotic pressure. Still others generate fragments that attract immune cells to the site of infection, serving as chemical beacons that say, "Come here, danger detected." The complement system is ancient, predating even the adaptive immune system, and it works with remarkable speed. Within seconds of detecting a threat, the cascade can be in full swing. It is a molecular chain reaction, a biological domino effect that amplifies a single recognition event into a powerful and coordinated response. Your immune system can detect and respond to an infection in a matter of hours. From the moment a pathogen breaches your defenses, the countdown begins. Resident immune cells in the affected tissue recognize molecular patterns associated with the invader. Fragments of bacterial cell walls, viral genetic material, fungal carbohydrates, and immediately begin secreting alarm signals called cytokines. These chemical messengers diffuse through the tissue and enter the bloodstream, alerting the entire body to the threat. Within minutes, blood flow to the area increases, bringing fresh supplies of oxygen and immune cells, while the walls of blood vessels become more permeable, allowing fluid and cells to leak into the tissue. Neutrophils arrive first, often within an hour, streaming out of the blood and into the infected area, where they begin attacking the invaders. If the infection is not quickly contained, dendritic cells carry samples of the pathogen to nearby lymph nodes, where they present their findings to T cells and B cells, initiating the slower but more powerful adaptive immune response. This rapid mobilization, this ability to detect trouble and respond with such speed, is what stands between you and countless infections that could otherwise overwhelm you. Inflammation is not an enemy, but a carefully controlled process of healing and repair. The redness, heat, swelling, and pain that characterize inflammation are often perceived as problems to be suppressed, but in truth, they are signs that your immune system is working exactly as it should. The redness and heat come from increased blood flow, which delivers oxygen, nutrients, and immune cells to the affected area. The swelling results from fluid leaking out of blood vessels, carrying antibodies and signaling molecules into the tissue where they are needed. The pain serves as a warning, encouraging you to protect the injured area while it heals. Inflammation is your body's way of walling off damage, preventing infection from spreading, clearing away debris, and orchestrating the reconstruction of tissue. It is a temporary state, designed to resolve once the threat has passed, and when it functions properly, it leaves you healthier than before, with the damage repaired and the lessons learned. Only when inflammation becomes chronic, persisting long after the initial threat is gone, does it become harmful, a fire that forgets to extinguish itself. Dendritic cells act as messengers, carrying information about threats from tissues to lymph nodes. These cells are the scouts and intelligence officers of the immune system, named for their branching, tree-like projections that reach out in all directions to sample their environment. Dendritic cells reside in tissues throughout the body, particularly at barrier sites like the skin, lungs, and gut, where they constantly collect samples of everything they encounter. When they capture a piece of a pathogen, a fragment of bacterial protein perhaps, or a viral particle, they undergo a remarkable transformation, maturing and migrating away from the tissue toward the nearest lymph node. This journey can take hours or days, the dendritic cell crawling through tissue and flowing through lymphatic vessels, carrying its precious cargo all the while. Upon reaching the lymph node, it presents its findings to naive T cells, displaying fragments of the pathogen on its surface like evidence laid out in a courtroom. This presentation is what initiates the adaptive immune response, the process by which your body learns to recognize and remember a specific threat. Without dendritic cells serving as this crucial link between tissues and lymph nodes, between innate detection and adaptive response, the immune system would be blind to much of what threatens it. Your immune system learns throughout your life, becoming more sophisticated with each exposure. Unlike many biological systems that peak in youth and decline with age, the adaptive immune system continues to grow wiser over the decades. Each infection, each vaccine, Each encounter with a novel antigen adds to the library of memory cells, expanding the repertoire of threats that can be recognized and rapidly countered. An elderly person's immune system may not respond as vigorously as a young person's, producing fewer new cells with each challenge, but it compensates with experience, a deeper catalogue of remembered enemies, a more nuanced understanding of self and non-self. This accumulation of immunological wisdom is why some infections that ravage the young spare the old, why travelers who grew up in regions with diverse pathogens often resist diseases that fell travelers from more sheltered environments. Your immune system is a student that never stops learning, an archive that grows richer with time, carrying forward the hard-won lessons of every battle survived. Regulatory T cells act as peacekeepers, preventing your immune system from attacking your own body. Among the T cell population, there exists a special subset whose job is not to attack, but to restrain, not to amplify the immune response, but to dampen it. These regulatory cells patrol your body, searching not for foreign invaders, but for immune responses that have become too aggressive, that threaten to cause more harm than the original infection. They secrete inhibitory signals that calm overactive immune cells, prevent inflammation from spiraling out of control, and maintain tolerance to your own tissues and to harmless substances like food and pollen. Without regulatory T cells, the immune system would turn on itself, attacking the very body it is meant to protect, leading to autoimmune disease and chronic inflammation. These peacekeepers are the voice of moderation, the wisdom that knows when enough is enough, when the battle has been won and it is time to stand down. They remind us that true power lies not only in the ability to fight, but also in the ability to show restraint. The lymphatic system has no pump. It relies on the gentle movement of your muscles and breathing. Unlike the cardiovascular system, which is driven by the powerful contractions of the heart, the lymphatic system depends on far subtler forces to maintain its flow. When your muscles contract during movement, they compress the lymphatic vessels that run through them, pushing lymph forward through a series of one-way valves that prevent backflow. When you breathe, the changing pressure in your chest cavity creates a gentle suction that draws lymph upward from the lower body. Even the pulsation of nearby arteries can help nudge lymph along its path. This means that prolonged immobility allows lymph to stagnate, which is why legs swell during long flights or periods of bed rest. Movement, even gentle movement, keeps the lymph flowing, keeps immune cells circulating, keeps the cellular debris and excess fluid draining from your tissues. The lymphatic system is a testament to the wisdom of motion, a reminder that your body is designed to move, that stillness for too long allows the waters within you to grow stagnant. Your appendix may act as a safe house where beneficial gut bacteria can hide during illness. Long dismissed as a vestigial organ with no real function, an evolutionary leftover prone to dangerous inflammation, the appendix has been reconsidered in recent years. This small, finger-like pouch, attached to the beginning of the large intestine, contains dense concentrations of immune tissue and seems to serve as a reservoir for beneficial bacteria. When severe diarrheal illness flushes the gut clean of its normal bacterial population, the appendix, tucked safely away from the main flow of intestinal contents, preserves a sample of the original community. Once the illness passes, bacteria can emerge from the appendix to recolonize the intestine, restoring the beneficial microbiome much more quickly than would occur through environmental exposure alone. It is a backup drive for your bacterial partners, a library that preserves the original text after the working copies have been destroyed. Not all scientists agree on this interpretation, but the hypothesis is elegant and plausible, suggesting that even structures we thought were useless may serve purposes we simply fail to recognize. IgA antibodies in breast milk pass immunity from mother to infant, a gift of protection. A newborn's immune system is naive, having encountered few, if any, pathogens in the sterile environment of the womb, and it will take months or years to build up its own catalogue of defenses. In the meantime, mother's milk provides temporary immunity, a loan of experience to bridge the vulnerable early period. IgA antibodies, specially designed to function in the harsh environment of the gut, coat the infant's digestive tract, neutralizing pathogens before they can establish infection. These antibodies are tailored to the mother's environment, reflecting the threats she has encountered and survived, which are likely to be the same threats the infant will face. Beyond antibodies, breast milk also contains immune cells, antimicrobial proteins, and factors that promote the growth of beneficial bacteria, creating a complete immunological package. This transfer of immunity is one of the first gifts a mother gives her child, a temporary shield that protects during those first precious months, while the infant's own defenses slowly awaken and mature. Your immune system can recognize and remember up to 1 billion different antigens. This number is almost incomprehensible, more different recognition patterns than there are people in entire continents, all encoded within the microscopic architecture of your immune cells. The diversity arises from genetic recombination, a process by which segments of DNA are randomly shuffled and joined during the development of each lymphocyte, creating a unique receptor unlike any other. The mathematical possibilities of this shuffling extend into the trillions, but practical constraints, cells that recognize your own tissues, must be eliminated, and the body can only maintain so many different cell types. Bring the functional repertoire down to roughly 1 billion distinct specificities. This still represents astonishing coverage of the molecular universe, ensuring that no matter what pathogen emerges, somewhere in your immune system, there likely exists a cell capable of recognizing it. It is a biological lottery where billions of tickets are purchased in advance, guaranteeing that one of them will match the winning numbers no matter what sequence is drawn. Neutrophils are the first responders, arriving at infection sites within minutes. These cells make up the majority of your white blood cells, comprising some 50 to 70% of all circulating leukocytes, and they exist for one primary purpose: rapid response. Neutrophils are short-lived, surviving only hours to days, but they are produced in such vast numbers that your bone marrow releases millions into your bloodstream every minute. When infection strikes, they are the first immune cells to arrive. Drawn by chemical signals from the site of damage, they squeeze out of blood vessels and crawl toward the source of infection, following the chemical gradient like moths drawn to flame. Once there, they attack with ferocity, engulfing bacteria, releasing antimicrobial chemicals, and even expelling their own DNA in sticky nets that trap and kill pathogens. Neutrophils often die in the process, sacrificing themselves in the line of duty, and the creamy substance we call pus is largely composed of dead neutrophils, the bodies of fallen soldiers. Their brief lives and violent deaths are the price of your protection, the front line that holds while reinforcements arrive. Your immune system works most actively while you sleep, repairing and consolidating defenses. Sleep is not merely a period of rest, but a time of intense immunological activity, when the body shifts resources from daytime functions like movement and cognition toward maintenance and defense. Studies have shown that certain immune cells peak in number during sleep, that the production of cytokines increases, and that the formation of immunological memory is enhanced during the sleeping hours. This is why illness so often disrupts sleep. The body is demanding the resources it needs to fight, and why sleep deprivation leaves you more vulnerable to infection. During sleep, your immune system also conducts surveillance, searching for cancer cells and dealing with the accumulated damage of the day. The immune system and the sleep cycle are intimately connected, each supporting the other, creating a rhythm of activity and rest that extends down to the cellular level. When you sleep, you are not absent, but deeply engaged in the work of maintaining yourself. Cytokines are chemical messengers that allow immune cells to communicate across vast distances. These small proteins are the words and sentences of the immune system's language, carrying information from cell to cell, from tissue to bloodstream, sometimes from one end of the body to the other. Some cytokines are alarm signals, announcing the presence of infection and calling for reinforcements. Others are instructions, telling nearby cells what type of response to mount or when to stand down. Still others promote inflammation, stimulate the production of new immune cells, or trigger fever by acting on the brain. A single immune cell can secrete dozens of different cytokines and can respond to dozens more, creating a communication network of staggering complexity. This molecular conversation happens continuously, a chemical symphony playing beneath your awareness, coordinating the activities of billions of cells into a unified response. Without cytokines, your immune system would be a collection of isolated soldiers rather than a coordinated army, unable to share information or work toward common goals. The blood-brain barrier protects your mind, allowing only select immune cells to enter. The brain is privileged territory, walled off from the general circulation by a specialized barrier formed by tightly joined cells that line the brain's blood vessels. This barrier exists because the brain is uniquely vulnerable. Its neurons cannot regenerate easily, and the inflammatory responses that heal damage elsewhere could cause catastrophic harm to neural tissue. Only small, select molecules can pass through the blood-brain barrier. An immune surveillance within the brain is conducted by specialized cells that are gentler and more restrained than their counterparts elsewhere. When infection or injury does breach this barrier, the response must be carefully calibrated, strong enough to address the threat, but not so vigorous that it destroys the delicate architecture of the mind. The brain exists in relative isolation, a fortress within a fortress, protected not only from the outside world, but from the very defenses that guard the rest of your body. Your tonsils and adenoids are strategic outposts, sampling pathogens that enter through your nose and mouth. Located at the entrance to the respiratory and digestive tracts, these masses of lymphoid tissue form a ring of immune surveillance around the airways, positioned to intercept threats as early as possible. Their surfaces are covered with crypts and folds that increase the surface area available for sampling, and they contain high concentrations of B cells, T cells, and other immune cells ready to respond to anything suspicious. When bacteria or viruses are inhaled or swallowed, they come into contact with the tonsils and adenoids, which capture samples and initiate an immune response. This is why these tissues sometimes swell and become inflamed. They are active battlegrounds. In children whose immune systems are still learning, the tonsils and adenoids are particularly large and active, serving as training grounds where the adaptive immune system encounters and learns to recognize common environmental pathogens. Though they can be removed without catastrophic consequences, their strategic positions suggest they play an important role in early immune defense. Eosinophils defend primarily against parasites, a reminder of our ancient evolutionary history. These specialized white blood cells are relatively rare in most healthy people, but they become abundant during parasitic infections, particularly those caused by worms and other multicellular invaders, too large to be engulfed by ordinary phagocytes. Eosinophils are packed with granules containing toxic proteins that can damage the outer surfaces of parasites, and they cluster around these invaders, releasing their chemical weapons in a coordinated assault. The persistence of eosinophils in the modern human immune system, even in populations where parasitic infections are now rare, speaks to the long history of our species' struggle against these ancient enemies. For most of human evolution, parasitic worms were a constant threat, and the immune system evolved specialized tools to combat them. Today, some scientists believe that the relative absence of parasites in developed nations may contribute to allergies and autoimmune diseases, as eosinophils and other anti-parasitic responses seek targets that no longer exist, turning instead against harmless substances like pollen and pet dander. Your immune system can distinguish between food proteins and dangerous pathogens in your gut. This is one of the immune system's most delicate balancing acts. The digestive tract is constantly exposed to foreign proteins from the food you eat, proteins that are harmless and even necessary for nutrition. And yet, it must remain vigilant against the genuinely dangerous bacteria and viruses that may contaminate that food. The gut immune system learns to tolerate dietary proteins through a process called oral tolerance, in which repeated exposure to a protein in the context of digestion trains immune cells to recognize it as safe rather than threatening. This education begins in infancy and continues throughout life, and it relies on specialized dendritic cells and regulatory T cells that promote tolerance rather than inflammation. When this process fails, the result can be food allergies or inflammatory bowel disease, conditions in which the immune system attacks what should be recognized as harmless. The wisdom of the gut immune system lies in its ability to make these fine distinctions, to know that the protein in an apple is different from the protein on the surface of a pathogen, even when both are foreign to the body. Mast cells release histamine, creating inflammation that brings more immune cells to threatened areas. These tissue-resident cells are stationed throughout the body, particularly abundant in the skin, lungs, and digestive tract, anywhere that interfaces with the external environment. Mast cells are packed with granules containing histamine and other inflammatory mediators, held ready like loaded weapons waiting for the signal to fire. When mast cells detect signs of tissue damage or infection, they degranulate, releasing their contents in a sudden burst that triggers a cascade of physiological changes. Histamine causes blood vessels to dilate and become more permeable, increasing blood flow to the area and allowing fluid and immune cells to leak into the tissue. This is what produces the characteristic redness, warmth, and swelling of inflammation, as well as the itching sensation that can accompany allergic reactions. While histamine release can be uncomfortable or even dangerous when it occurs inappropriately, as in severe allergies, its purpose is fundamentally protective, to summon reinforcements and create conditions that favor immune defense and healing. The adaptive immune system appeared over 500 million years ago in the first jawed fish. Before this pivotal moment in evolutionary history, organisms relied solely on innate immunity. pattern recognition receptors and phagocytic cells that could respond to general features of pathogens but could not learn or remember specific threats. Then, in an ancestral jawed fish swimming in ancient seas, a remarkable innovation emerged. Lymphocytes with randomly generated receptors capable of recognizing specific antigens and the ability to remember those antigens for the lifetime of the organism. This leap forward likely arose from the accidental insertion of mobile genetic elements into the genes that encode immune receptors, creating the machinery for recombination that still operates in your immune system today. From those first jawed fish, the adaptive immune system passed to amphibians, reptiles, birds, and mammals, refining and elaborating over hundreds of millions of years, but retaining the same fundamental mechanisms. When your B cells shuffle gene segments to create new antibodies, they are using molecular machinery that first appeared in creatures that lived 500 million years ago. A genetic inheritance older than forests, older than flowers, older than bones themselves. Your immune system creates new antibodies by randomly shuffling genetic segments like a molecular deck of cards. The genes that encode antibody receptors are not inherited as complete functional units, but as scattered segments. Dozens of V segments, several D segments, multiple J segments, and various C segments that must be assembled during the development of each B cell. Enzymes cut and paste these segments together in random combinations, joining one V to one D to one J, creating a unique sequence that will determine what antigen that particular B cell can recognize. The randomness of this process means that each B cell develops a different receptor, and together they create a repertoire vast enough to recognize almost any molecular shape. Further diversity is added through imprecise joining, which inserts or deletes nucleotides at the junction points, and through somatic hypermutation, which introduces random changes to the antibody genes during immune responses, allowing the antibodies to evolve toward better binding. This deliberate introduction of randomness and mutation into the genome, usually so carefully guarded against change, represents a calculated risk, a sacrifice of genetic stability in exchange for the flexibility needed to defend against an unpredictable microbial world. Plasma cells are antibody factories, producing thousands of identical antibodies every second. When a B cell encounters its specific antigen and receives the proper activation signals, it can transform into a plasma cell, abandoning all other functions in favor of a single purpose: mass production of antibodies. The plasma cell's internal architecture reorganizes, expanding the endoplasmic reticulum, the cellular machinery responsible for protein synthesis, until it fills most of the cell's volume. The cell swells with the effort, becoming engorged with antibody molecules in various stages of assembly, and secretes them into the surrounding fluid in a continuous stream. A single plasma cell can produce 2,000 antibody molecules per second, up to 10 million per hour, maintaining this furious pace for days or weeks until it exhausts itself and dies. These cells are the infantry of the adaptive immune response, short-lived and specialized, flooding your body with the specific antibodies needed to neutralize a particular threat. They are the reason that antibody levels rise sharply during infection, reaching concentrations high enough to coat every viral particle, to neutralize every toxin molecule, to mark every bacterial cell for destruction. Your immune system ages with you, gradually becoming less responsive over decades. This process, called immunosenescence, is a slow dimming of the immune system's light, a gradual loss of the vigor and flexibility that characterized youth. The thymus, which trains new T cells, shrinks with age, producing fewer fresh recruits to join the immune ranks. Existing immune cells become less responsive to activation signals, slower to divide, less efficient at clearing infections. The production of new antibodies declines, and the memory cells that accumulated over a lifetime begin to dominate the immune repertoire, leaving fewer resources for responding to novel threats. This aging is not failure, but rather a shifting of priorities. The body's investment in growth and reproduction gives way to maintenance and preservation. Yet even in advanced age, the immune system continues its work, drawing on the deep well of experience accumulated over decades, fighting with wisdom if not with youthful speed. The aged immune system is a veteran warrior, scarred and slower but rich in knowledge that only time can provide. Gut bacteria help train your immune system, teaching it to recognize friend from foe. From the moment of birth, when you are first colonized by microbes from your mother and the environment, your immune system begins a lifelong education in discrimination. The bacteria that live in your gut, numbering in the trillions, representing thousands of different species, constantly interact with immune cells in the intestinal wall, sending signals that calibrate the immune response. Some bacteria promote the development of regulatory T cells, teaching tolerance and restraint, while others stimulate the production of antimicrobial peptides or train immune cells to recognize common pathogenic patterns. Mice raised in completely sterile environments with no bacterial exposure at all develop stunted and dysfunctional immune systems, proving that this microbial education is not optional but essential. Your bacteria are not merely passengers, but partners, ancient allies whose ancestors have co-evolved with ours for millions of years, shaping the development and function of immunity itself. Your immune cells communicate using the same neurotransmitters as your brain: serotonin, dopamine, and more. This shared chemical vocabulary reveals a deep connection between the nervous system and the immune system. Two networks that were once thought to operate independently, but are now understood to be in constant dialogue. Immune cells carry receptors for neurotransmitters, allowing them to sense and respond to signals from nerves, while immune cells themselves can produce these same chemicals, potentially influencing mood and behavior. Serotonin, best known for its role in regulating mood and sleep, also modulates immune cell migration and the intensity of inflammatory responses. Dopamine can suppress certain immune functions, perhaps explaining why stress and depression are associated with increased susceptibility to infection. This biochemical overlap suggests that mind and body are not separate domains, but aspects of a unified system, where thoughts can influence immunity and where immune activity can shape the landscape of consciousness itself. The spleen holds a reserve of immune cells, ready to release them during emergencies. Beyond its role as a filter and surveillance post, the spleen serves as a strategic reserve, a biological depot where monocytes and other immune cells are stored in higher concentrations than anywhere else in the body. During times of severe stress or injury, a heart attack, a traumatic wound, a sudden overwhelming infection, the spleen contracts, squeezing these stored cells into circulation and boosting the available immune response by as much as 40%. This reservoir function is particularly important for monocytes, which can quickly mature into macrophages and migrate to damaged tissue to promote healing and clear debris. The spleen's ability to modulate the size of the immune force releasing reinforcements when needed and storing them when not, represents another layer of the immune system's sophistication, a capacity for rapid mobilization that can make the difference between surviving and succumbing to sudden threats. Your immune system can detect the chemical signatures of stress hormones and adjust its response. When you experience stress, whether physical or psychological, your body releases hormones like cortisol and adrenaline, and these molecules do not merely affect your heart rate and mental state, but also communicate directly with immune cells. In the short term, stress can enhance immune function, preparing the body for potential injury by mobilizing immune cells and sharpening inflammatory responses. But when stress becomes chronic, the constant presence of these hormones begins to suppress immune activity, reducing the production of new immune cells, inhibiting the function of existing ones, and promoting a shift toward inflammation that is less helpful and more harmful. The immune system, in this way, is exquisitely sensitive to your emotional and psychological state, responding to the chemical echoes of your experiences and adapting its behavior accordingly. This connection explains why chronic stress leaves you vulnerable to illness, why grief can manifest as physical sickness, why the state of your mind shapes the strength of your defenses. Langerhans cells in your skin act as sentinels, capturing antigens and carrying them to lymph nodes. Named after the German physician who first described them in 1868, these dendritic cells form a dense network throughout the epidermis, extending their branching processes between skin cells to constantly sample the environment. Their position at the body's outer boundary makes them early detectors of any breach, a cut, a puncture, an infection entering through broken skin. When Langerhans cells encounter foreign antigens, they undergo the same transformation as other dendritic cells, maturing and migrating away from the skin toward the nearest lymph node, where they will present their findings to T cells. This journey from skin to lymph node can take 1 to 2 days, the Langerhans cell traveling through tissue and lymphatic vessels, crossing the boundary between the external world and the internal immune architecture. They are the watchers on the wall, the scouts who bring news from the frontier, initiating adaptive responses to threats that have breached the first barrier. Your immune system removes about 200 billion red blood cells every day, recycling their components. Red blood cells, which carry oxygen throughout your body, have a lifespan of approximately 120 days, after which they become old and damaged, their membranes less flexible, their function declining. Macrophages in the spleen, liver, and bone marrow recognize these aging cells and remove them from circulation, breaking them down in a process of careful disassembly. The iron from hemoglobin is salvaged and returned to the bone marrow to be incorporated into new red blood cells, while the protein components are broken down into amino acids that can be reused elsewhere, and the heme groups are converted into bilirubin, which is eventually excreted. This recycling operation runs constantly and efficiently, removing roughly 200 billion cells per day, a number almost incomprehensible in its scale, while replacing them with an equal number of fresh cells. It is demolition and construction happening simultaneously, a perfect balance that keeps your blood young even as individual cells age and die. B cells can produce up to 2,000 antibody molecules per second at peak production. When a plasma cell dedicates itself entirely to antibody synthesis, it becomes one of the most productive cells in the body, its internal machinery running at maximum capacity. The endoplasmic reticulum, where proteins are folded and assembled, expands to fill most of the cell's volume, and ribosomes, the molecular machines that translate genetic instructions into protein, coat its surface in dense clusters. Chaperone proteins work frantically to ensure each antibody is properly folded, while quality control mechanisms check for errors and discard malformed molecules. The cell becomes a factory floor running 3 shifts around the clock, never pausing, never resting, producing antibody after antibody in an endless stream. This extraordinary rate of production, 2,000 molecules per second, millions per hour, is what allows antibody levels to rise so quickly during infection, creating concentrations high enough to neutralize the threat before it can overwhelm the body's other defenses. The immune system has its own circadian rhythm, being more active at certain times of day. Just as your sleep and wakefulness follow a daily cycle, so too does your immune system, with certain functions peaking in the evening and night, while others are more active during the day. The number of immune cells in circulation varies across the 24-hour cycle, with some types being more abundant during rest and others during activity. Inflammatory responses tend to be stronger in the early morning hours, which may explain why asthma attacks and heart attacks often occur at dawn. The timing of immune activity is coordinated by the same molecular clocks that regulate sleep, hunger, and body temperature, all synchronized to the daily rotation of the Earth. This circadian variation has practical implications. Vaccines may be more effective when administered at certain times of day, and the timing of infection can influence how severe the resulting illness becomes. Your immune system is not constant, but rhythmic, pulsing with the same daily beat that governs all of life. Mucus traps pathogens like flypaper, preventing them from reaching vulnerable cells beneath. The sticky, viscous layer that coats your respiratory and digestive tracts is far more than an annoyance. It is a highly sophisticated defense system. Mucus is composed of water, salts, and large glycoproteins called mucins that form a gel-like mesh capable of trapping particles and microbes. Anything that lands on this surface becomes stuck, unable to reach the epithelial cells below where it might establish infection. The mucus layer is not static, but constantly moving, swept along by the coordinated beating of cilia, tiny hair-like projections that cover the surface of respiratory cells and move in synchronized waves. This mucociliary escalator carries trapped particles and pathogens up and out of the lungs toward the throat, where they can be coughed up or swallowed into the stomach's acidic depths. It is a conveyor belt of protection, continuously clearing away threats before they can take hold, operating every moment without your awareness or effort. Your immune system can detect cancer cells by recognizing abnormal proteins on their surface. As cells become cancerous, they undergo changes that mark them as different, as no longer quite self. They may display mutated proteins or express proteins that are normally only present during fetal development, or lose the identification molecules that mark them as belonging. Natural killer cells and cytotoxic T cells patrol constantly, scanning the surfaces of the cells they encounter for these telltale signs of transformation. When they find a cell that has become cancerous, they can destroy it before it multiplies, preventing a tumor from ever forming. This process, called immune surveillance, is thought to eliminate countless potential cancers throughout your lifetime, a silent culling that happens without your knowledge. Cancer develops only when transformed cells manage to evade or suppress this surveillance, acquiring mutations that make them invisible to immune detection or that actively inhibit the immune response. The immune system's role in cancer prevention is both a source of hope, suggesting that enhancing immune surveillance might prevent or treat cancer, and a reminder of how remarkable it is that cancer remains relatively rare, given the trillions of cell divisions that occur in a human lifetime. Interferon is a protein that warns neighboring cells of viral infection, allowing them to prepare defenses. When a cell becomes infected with a virus, one of its first responses is to produce and secrete interferon, a small protein that diffuses to nearby cells and binds to receptors on their surface. This binding triggers a cascade of changes within the receiving cell. Hundreds of genes are activated, producing proteins that interfere with viral replication, degrade viral genetic material, and make the cell less hospitable to viral takeover. Interferon also activates nearby immune cells, enhancing their ability to detect and destroy infected cells. The name interferon reflects this function. It interferes with the virus's ability to spread from cell to cell, buying time for the adaptive immune response to develop. Different types of interferon exist, each specialized for different contexts, but all share this fundamental role as an early warning system, a chemical alarm that spreads through tissue faster than the virus itself can travel. The immune system uses inflammation to wall off infections, preventing them from spreading. When a localized infection threatens to spread beyond its initial site, the immune system can create a physical barrier around it through a process called granuloma formation or abscess formation. Immune cells and inflammatory signals cause blood vessels to leak fluid and proteins into the surrounding tissue, creating a zone of swelling and increased pressure that is difficult for bacteria to penetrate. Fibrin, the same protein that forms blood clots, can be deposited in a mesh around the infected area, creating a physical wall that contains the infection. Immune cells cluster densely at the boundary, attacking any pathogens that attempt to escape while preventing the spread of infection into healthy tissue. This strategy of containment is ancient and effective, sacrificing a small area of tissue to protect the whole, creating a battlefield that is deliberately isolated from the rest of the body. The walling off of infection is yet another example of the immune system's strategic sophistication, its ability to make calculated decisions about where to fight and where to retreat. Your Peyer's patches in the intestine sample gut contents, monitoring for dangerous invaders. These specialized structures, named after the Swiss anatomist who described them in the 17th century, are clusters of lymphoid tissue embedded in the wall of the small intestine. Their surfaces are covered with specialized cells called M cells, which actively sample the contents of the gut, transporting small amounts of bacteria, food particles, and other antigens from the intestinal lumen across the barrier and presenting them to immune cells waiting on the other side. Peyer's patches are training grounds where the gut immune system learns what is normal, what is beneficial, and what is dangerous. They contain dense collections of B cells and T cells, and it is here that many immune responses to gut pathogens are initiated. The constant sampling and assessment that occurs in Peyer's patches is what allows your immune system to maintain the delicate balance between tolerance and defense in an environment teeming with trillions of bacteria. NK cells can kill infected cells in minutes by injecting enzymes that trigger self-destruction. Natural killer cells do not need to wait for activation or instruction. They are born ready to kill, constantly patrolling and assessing every cell they encounter. When an NK cell identifies a target, a cell infected with virus or transformed by cancer, it forms a tight connection with that cell and releases the contents of specialized granules directly into the space between them. These granules contain perforin, a protein that punches holes in the target cell's membrane, and granzymes, enzymes that enter through these holes and trigger apoptosis, the target cell's own self-destruction program. Within minutes, the target cell begins to die in an orderly fashion, breaking itself down into small packages that can be cleared away without causing inflammation or damage to surrounding tissue. The NK cell then detaches and moves on, searching for its next target, a serial killer working in service of your survival. This rapid, efficient elimination of compromised cells prevents infections from spreading and tumors from growing, a form of quality control that operates continuously throughout your body. Your immune system maintains tolerance to trillions of beneficial bacteria living on and in your body. The bacteria that colonize your skin, mouth, gut, and other surfaces outnumber your own cells, and yet your immune system does not attack them. This tolerance is not passive ignorance, but active recognition and accommodation, a learned acceptance that develops through constant interaction. Regulatory immune cells in barrier tissues suppress inflammatory responses to these beneficial bacteria, while the bacteria themselves produce signals that promote immune tolerance and help maintain the barrier's integrity. The relationship is symbiotic. The bacteria gain a stable home and steady supply of nutrients, while you gain assistance with digestion, production of vitamins, protection against pathogens that might try to colonize the same spaces, and training for your immune system. When this tolerance breaks down, as can happen after antibiotic treatment or in inflammatory bowel disease, the immune system may begin attacking the beneficial microbiome, creating inflammation that harms the host more than any pathogen could. The ability to coexist peacefully with this vast microbial community is one of the immune system's most impressive achievements. The lymphatic system drains 3 liters of fluid from your tissues back into your bloodstream every day. Blood pressure forces fluid out of capillaries and into the spaces between cells. And while most of this fluid is reabsorbed into the blood vessels, a significant amount remains behind. Without the lymphatic system to collect this excess fluid and return it to circulation, your tissues would swell with accumulated water, pressure would build, and the delicate balance of your internal environment would collapse within hours. The lymphatic vessels act as a drainage system, collecting the leaked fluid along with any proteins, cellular debris, and pathogens it has picked up, and channeling it through a series of filtration stations, the lymph nodes, before returning it to the bloodstream. 3 liters per day, every day, collected and returned in a circuit that runs parallel to your cardiovascular system, a shadow circulation that is just as essential but far less celebrated. Without this constant drainage, the blood volume would drop, tissue pressure would rise, and life would become impossible. Granulocytes contain granules filled with antimicrobial chemicals, ready to release on command. These cells, neutrophils, eosinophils, and basophils, are named for the distinctive granules visible under a microscope. Granules that give them a speckled appearance and contain some of the most destructive chemicals in your body. The granules are packets of enzymes, reactive oxygen species, and antimicrobial peptides, all held safely contained until the moment they are needed. When a granulocyte encounters a pathogen or receives the appropriate activation signal, it can release these granules either inside itself after engulfing the target, or externally into the surrounding tissue. The chemicals inside are powerful enough to break down bacterial cell walls, degrade proteins, and kill microbes through oxidative damage, but they are also dangerous to your own tissues if released inappropriately. Granulocytes must balance their destructive potential with restraint, unleashing their weapons only when and where they are needed, walking the line between insufficient response and collateral damage. Your immune system creates a chemical memory of smells associated with illness, helping you avoid them. There is a reason why certain smells can trigger an immediate sense of revulsion or nausea, why the odor of spoiled food can make you recoil before you've consciously identified the source. Your immune system, working in concert with your nervous system, forms associations between olfactory cues and past illnesses, creating learned aversions that protect you from repeating dangerous exposures. When you become sick after eating a particular food, your brain and immune system together encode that experience, linking the smell of that food with the physiological distress that followed. This conditioned taste aversion is so powerful that it can persist for years, even when you consciously know the food was not actually responsible for the illness. It is an ancient protective mechanism, a form of learning that operates below the level of conscious thought, steering you away from potential dangers through the simple language of disgust and attraction. Basophils are the rarest white blood cells, but play crucial roles in allergic responses. Comprising less than 1% of circulating white blood cells, basophils are so rare that they were once thought to be insignificant, but research has revealed them to be key players in certain immune responses. Like their tissue-dwelling relatives, the mast cells, basophils contain granulase packed with histamine and other inflammatory mediators, and they release these chemicals during allergic reactions and parasitic infections. They are among the first cells to respond to allergens, helping to initiate the cascade of inflammation that characterizes allergic responses. Basophils also produce cytokines that influence other immune cells, shaping the broader immune response in ways that scientists are still working to fully understand. Their rarity makes them difficult to study, but their importance is increasingly recognized. They are not merely bystanders, but active participants in immune defense and sometimes in immune-mediated disease. Your immune system can learn to tolerate allergens through repeated, controlled exposure. This principle underlies immunotherapy, a treatment in which people with severe allergies are given gradually increasing doses of the substance they are allergic to, training their immune system to accept it rather than attack it. Over weeks or months of carefully controlled exposure, regulatory T cells learn to recognize the allergen as harmless, suppressing the inflammatory response that previously caused symptoms. The immune system's flexibility, its capacity to learn and relearn throughout life, makes this therapeutic approach possible. What was once treated as an enemy can, with patience and repetition, become recognized as neutral or even beneficial. This plasticity is a double-edged sword. It allows the immune system to adapt to changing environments and recover from inappropriate responses. But it also means that tolerance can be lost if exposures cease, requiring ongoing maintenance. The immune system is not fixed but dynamic, capable of revision and reinterpretation throughout your life. Complement proteins can punch holes in bacterial cell walls, causing them to burst like balloons. The membrane attack complex, formed by several complement proteins assembling together, is a molecular weapon of elegant simplicity. These proteins insert themselves into the bacterial cell membrane and form a ring-shaped pore that allows water and ions to flow freely in and out. Bacteria, like most cells, maintain a careful balance of salts and water across their membranes. And disrupting this balance is catastrophic. Water rushes into the bacterium following the concentration gradient, and the cell swells, unable to control its internal pressure. Within seconds or minutes, the bacterial membrane ruptures, spilling the cell's contents into the surrounding fluid, and the bacterium dies. This mechanism is effective against many types of bacteria, particularly those with exposed membranes. Though some bacteria have evolved defenses against complement attack, coating themselves with protective molecules or actively destroying complement proteins before they can assemble. The evolutionary arms race between immune defenses and bacterial countermeasures has been ongoing for hundreds of millions of years, driving innovation on both sides. Your immune system produces more than 100 million different types of antibodies throughout your life. While the theoretical diversity of antibodies is measured in the trillions, the practical repertoire that actually exists in your body at any given time is smaller, but still staggering. At least 100 million distinct antibody specificities present simultaneously. This number reflects the compromise between maximal diversity and limited resources. Your body can only maintain so many different B-cell clones, only produce so many different antibodies before the cost becomes prohibitive. But 100 million is still enough to cover the vast majority of potential threats, ensuring that almost any pathogen that emerges will encounter an antibody capable of recognizing it. Over your lifetime, as you encounter new antigens and generate new responses, the composition of your antibody repertoire shifts and evolves, favoring those specificities that have proven useful, while allowing unused specificities to fade. Your antibody repertoire is a living library, constantly updating its catalog to reflect the world you inhabit. Immune cells migrate toward infection sites by following chemical gradients, like following a scent. This process, called chemotaxis, is how immune cells navigate the complex landscape of your body to find the sites where they are needed. Infected or damaged tissues release chemicals called chemokines, attractant molecules that diffuse outward from the source, creating a concentration gradient. Immune cells carry receptors that detect these chemokines, and when a cell senses that the concentration is higher on one side than the other, it moves in the direction of increasing concentration, climbing the gradient toward its source. It is navigation by smell, a molecular scent trail that guides cells through tissue toward infection. Different chemokines attract different cell types, allowing the immune system to recruit specific reinforcements. Neutrophils early, monocytes later, lymphocytes when adaptive immunity is needed. The elegance of this system lies in its simplicity. No central coordinator is required, no map or directions, just a chemical signal and cells programmed to follow it. Your thymus shrinks with age, but continues producing some new T cells even in old age. The thymus reaches its maximum size during childhood, when the need for new T cells is greatest and the body is building its initial catalogue of immune defenses. After puberty, it begins a slow, progressive shrinkage, gradually being replaced by fatty tissue until only a small remnant of functional thymic tissue remains in the elderly. This involution was once thought to represent the complete cessation of T-cell production, but research has revealed that even a very small thymus continues to educate and release new T-cells, albeit at a much reduced rate. The decline in thymic function is one of the primary drivers of immunosenescence, contributing to the reduced ability of older individuals to respond to novel pathogens and vaccines. Yet the fact that the thymus continues working at all, decades after its peak, speaks to the importance of maintaining at least some capacity for generating new, naive T cells, capable of recognizing threats never before encountered. The aging thymus is a reminder that biological systems rarely shut down completely, but rather fade gradually, maintaining function at reduced levels, even as resources are redirected elsewhere. The immune system removes over 50 billion cells from your body every day through programmed cell death. This enormous daily culling includes not only aged red blood cells, but also white blood cells that have completed their tasks, cells that are damaged or no longer needed, and cells that pose a potential threat. Apoptosis, programmed cell death, is a carefully controlled process in which cells dismantle themselves from within, breaking down their components in an orderly fashion and packaging the debris for easy removal. Unlike necrotic death, which is messy and inflammatory, apoptotic death is quiet and clean, designed to eliminate cells without causing collateral damage. Phagocytic cells patrol constantly, recognizing and engulfing apoptotic cells before they can rupture and spill their contents. This daily elimination of billions of cells is essential for maintaining balance, for preventing the accumulation of damaged or potentially dangerous cells, and for making room for the constant influx of new cells from bone marrow and other sources. Your body is not a static structure, but a river of cells, continuously flowing, continuously renewed. Antibodies can remain in your blood for months after an infection, providing ongoing protection. Even after the infection has been cleared and the plasma cells that produce the antibodies have died, the antibodies themselves persist, floating through your blood and lymph like sentinels left behind to guard against the invader's return. Different classes of antibodies have different lifespans. Some persist for only weeks, while others can remain for many months. But all eventually break down and are cleared from circulation. During this period of antibody persistence, you retain a measure of immediate protection against reinfection, a temporary immunity that bridges the gap between acute infection and long-term immunological memory. If the pathogen returns during this window, the antibodies can neutralize it immediately, often preventing symptoms entirely. Only after the antibodies have decayed does your protection rely solely on memory cells, which must be reactivated and must take several days to produce new antibodies. This lingering antibody presence is why recent infections provide stronger protection than distant ones, why boosters are sometimes needed years after initial vaccination. Your immune system can distinguish between identical twin cells that have become cancerous. Even though cancer cells arise from your own tissues and carry your own DNA, the process of transformation introduces changes, mutations in key genes, altered patterns of protein expression, the appearance of abnormal or embryonic proteins that mark them as aberrant. Immune cells, particularly cytotoxic T cells and natural killer cells, can detect these subtle differences, recognizing that something has gone wrong even when the cell is still overwhelmingly self. This discrimination is incredibly fine-grained. Two cells that were once identical, one of which has become cancerous, present differently enough for the immune system to identify and target the cancer cell while sparing the healthy one. The challenge lies in the fact that cancer cells evolve under immune pressure, selecting for mutations that make them less visible or more resistant to immune attack, a process called immunoediting. The immune system shapes the evolution of cancer, eliminating the most visible cells and inadvertently selecting for those that can hide or fight back, creating a dynamic struggle that can last for years. Lymphocytes can live for years, decades, or even your entire lifetime, depending on their type. While most immune cells are short-lived, measured in hours or days, lymphocytes, the B cells and T cells of adaptive immunity, exist on a different timescale. Some lymphocytes are effector cells, designed to mount an immediate response and then die after days or weeks. But others are memory cells, designed to persist indefinitely. Memory T cells and memory B cells can survive for decades, maintained by occasional cell division and by signals from the environment that promote their survival. There may be memory cells in your body right now that formed in childhood, that remember infections from 50 or 60 years ago, that have outlasted friendships and careers and homes. These long-lived cells are your immunological continuity, the thread that connects your past exposures to your present defenses, ensuring that the lessons learned over a lifetime are never entirely forgotten. The immune response generates heat as a byproduct of increased cellular activity. When immune cells activate, divide, migrate, and attack pathogens, they consume energy at a dramatically increased rate, burning glucose and oxygen to fuel their activities. This metabolic surge generates heat, just as exercise generates heat in muscles, contributing to the warmth you feel during fever and inflammation. The heat is not merely a useless byproduct, but may contribute to the antimicrobial effects of the immune response, creating local conditions that are less favorable for pathogen growth while supporting the accelerated activity of immune cells. At the site of infection or inflammation, the tissue can be measurably warmer than surrounding areas, a local fever that supplements the systemic fever generated by the hypothalamus. This thermal signature is why infected areas feel hot to the touch, why inflammation brings warmth as well as redness and swelling, and why your whole body may feel feverish during a significant immune response. Your immune system learns to ignore food proteins in the gut while remaining vigilant for pathogens. This dual awareness, tolerance for nutrition, vigilance against danger, is calibrated through constant exposure and specialized regulatory cells that dampen responses to harmless dietary proteins. The gut immune system samples everything you eat, learning to distinguish the molecular signature of food from the patterns associated with infection. It is a delicate equilibrium, maintained meal by meal, allowing you to extract nourishment from the world without treating every bite as an invasion. Microfold cells in your intestine transport antigens to immune cells for sampling and analysis. These specialized cells, scattered throughout the intestinal lining, actively capture particles from the gut and ferry them across the barrier to waiting immune cells below. They are windows into the digestive world, allowing the immune system to monitor what passes through without needing to breach the protective barrier. Through these cellular portals, your body learns what inhabits your gut, building knowledge one sample at a time. The immune system uses apoptosis, programmed cell death, to remove infected cells without causing inflammation. When a cell is infected with virus, triggering its self-destruction eliminates both the cell and the pathogen it harbors, all while avoiding the collateral damage of inflammatory death. The cell quietly dismantles itself, packages its components neatly, and is cleared away without alarm or disruption. It is a surgical strike, precise and contained, sacrificing individual cells to preserve the greater whole. Your immune cells can recognize patterns associated with bacterial DNA that differ from your own. Bacterial DNA contains unmethylated CpG sequences, patterns that are rare in mammalian DNA, and immune cells carry receptors specifically designed to detect these molecular signatures. This recognition allows the immune system to identify bacterial invaders based on their genetic material alone, even before they cause visible damage. It is pattern recognition at the molecular level, distinguishing friend from foe by reading the language written in DNA itself. High endothelial venules in lymph nodes are specialized gateways where lymphocytes enter from the blood. These unique blood vessels have thick cuboidal walls, unlike the flat cells lining ordinary capillaries, and they express adhesion molecules that allow lymphocytes to exit the bloodstream and enter the lymph node. They are immigration checkpoints, portals through which immune cells pass from circulation into the filtering stations where they will screen for threats. Without these specialized gateways, lymphocytes could not efficiently patrol the lymphatic system. The immune system creates germinal centers in lymph nodes, training grounds where B cells improve their antibodies. Within these microscopic structures, B cells undergo rapid mutation and selection, testing thousands of antibody variants to find those that bind most tightly to the antigen. It is evolution in miniature, compressed into days rather than generations, producing antibodies that are progressively refined toward perfection. The best performers survive and multiply, while those that fail are eliminated, creating antibodies exquisitely tailored to their targets. Your immune system can mount different types of responses depending on the specific threat detected. Intracellular viruses trigger one pattern of response, extracellular bacteria another, parasitic worms yet another, each requiring different combinations of cells, antibodies, and inflammatory signals. The immune system reads cues from the pathogen, its location, its molecular patterns, the damage it causes, and selects the appropriate counter-strategy. It is tactical flexibility, the ability to fight each enemy with the weapons most likely to succeed. Opsonization is when antibodies code pathogens, marking them for destruction by other immune cells. The word comes from the Greek for to prepare food, and the analogy is apt. Antibodies make pathogens more palatable to phagocytic cells, coating them in molecular sauce that says, "Eat me." This marking dramatically increases the efficiency of phagocytosis, turning pathogens that might have been ignored into irresistible targets. It is teamwork at the molecular level, antibodies and phagocytes working in concert. Your immune system maintains a careful balance between activation and suppression. Too little activity leaves you vulnerable to infection, while too much causes autoimmunity and chronic inflammation. Regulatory cells constantly monitor and adjust the immune response, amplifying it when threats are present and dampening it when danger has passed. This dynamic equilibrium is maintained through countless feedback loops, a biological thermostat that keeps immune function within safe and effective boundaries. Toll-like receptors recognize ancient molecular patterns associated with bacteria, viruses, and fungi. These receptors, found on many immune cells, detect structures that are common to broad classes of pathogens but absent from human cells. Bacterial flagella, viral RNA, fungal cell walls. When a toll-like receptor binds its target, it triggers immediate defensive responses, sounding an alarm that mobilizes the innate immune system. They are the earliest warning system, detecting danger through recognition of patterns preserved across millions of years of microbial evolution. The immune system can learn from vaccines, which teach it to recognize threats it has never encountered. A vaccine presents a safe version of a pathogen, killed, weakened, or fragmented, allowing your immune system to study it, generate antibodies, and create memory cells without the danger of actual infection. When the real pathogen arrives, your body responds as though remembering an old enemy, mounting a swift and effective defense. Vaccines are rehearsals for battles not yet fought, preparation encoded in molecular memory. your immune cells produce reactive oxygen species, controlled bursts of chemical destruction. These highly reactive molecules, the same chemistry that causes metal to rust, are weaponized by phagocytes to destroy engulfed pathogens. The burst of oxidative power can shred bacterial proteins, rupture membranes, and damage DNA, all contained within the protective bubble of a phagosome. It is controlled fire, destruction harnessed and directed with precision. Lymph flows slowly through lymph nodes, giving immune cells time to screen for threats. Unlike blood, which rushes through the heart-driven circulation, lymph percolates gently through the spongy architecture of lymph nodes, allowing resident immune cells to examine every drop. This deliberate slowness is strategic, ensuring that no pathogen passes through undetected. The lymph node is both filter and meeting ground, where fluid surveillance meets cellular intelligence. The immune system coordinates repair after injury, orchestrating inflammation and tissue regeneration. Immune cells do not merely fight infection, but also direct healing, secreting growth factors that stimulate cell division, signals that attract repair cells, and enzymes that remodel damaged tissue. Macrophages, in particular, transition from destroying pathogens to promoting reconstruction, changing their function as the battlefield becomes a construction site. The immune system is both warrior and builder. Your immune system can sense damage to your own cells, even without infection. When cells are injured by trauma, toxins, or stress, they release internal molecules that normally never appear outside the cell, and these damage-associated molecular patterns trigger immune responses just as pathogens do. The immune system recognizes not only foreign invasion, but also internal distress, responding to sterile injury with the same vigilance it shows toward infection. Follicular dendritic cells preserve antigens for months, helping maintain immunological memory. Unlike other dendritic cells that present antigens briefly and then die, follicular dendritic cells capture antigens on their surfaces and hold them indefinitely, displaying them to passing B cells in lymph node germinal centers. This long-term antigen display allows memory B cells to periodically revisit the original threat, refreshing their training and maintaining their readiness. The immune system uses redundancy. Many different cells and molecules can perform similar functions. If one pathway fails, others can compensate, ensuring that no single point of failure can completely disable your defenses. This biological backup system makes the immune response robust and resilient, able to function even when components are missing or damaged. It is engineering through evolution, reliability through duplication. Your immune cells can change their behavior based on chemical signals from other cells. A macrophage can shift from inflammatory to anti-inflammatory. A T cell can change from helper to suppressor, all depending on the cytokine environment they encounter. This plasticity allows the immune system to adapt dynamically to changing circumstances, fine-tuning its response in real time. Marginal zone B cells respond rapidly to blood-borne pathogens before they can spread. Stationed at the border between the red pulp and white pulp of the spleen, these specialized B cells encounter pathogens as soon as they enter the bloodstream and can produce antibodies within hours, bridging the gap between innate and adaptive immunity. They are the rapid response force, buying time for more refined defenses to develop. The immune system creates physical barriers, chemical defenses, and cellular armies, a multi-layered shield. Skin blocks entry, mucus traps invaders, antimicrobial peptides kill on contact, phagocytes engulf and destroy, and lymphocytes remember and target with precision. Each layer compensates for the limitations of the others, creating defense in depth. No single barrier is impenetrable, but together they form a protection that is difficult to breach. Your immune system is connected to your nervous system, allowing mind and body to communicate. Nerve endings release neurotransmitters that influence immune cell behavior, while immune cells produce cytokines that signal the brain, affecting mood, sleep, appetite, and cognition. The two systems are in constant dialogue, each shaping the other's activity. Stress dampens immunity, while illness triggers behavioral changes, both reflections of this deep integration. Tissue-resident memory cells remain in specific organs, providing rapid local protection. Rather than circulating through blood and lymph, these specialized T cells take up permanent residence in tissues like the skin, lungs, and gut, standing guard at the sites most likely to encounter repeat infections. They can mount defenses within hours of pathogen arrival, faster than any circulating cell could respond. The immune system can suppress its own activity to prevent damage to healthy tissue. As powerful as immune defenses are, they carry the potential for collateral damage, inflammation that destroys healthy cells along with pathogens, bodies that might cross-react with self-proteins. Regulatory mechanisms act as brakes, releasing anti-inflammatory signals that calm overactive responses and prevent the immune system from becoming its own worst enemy. This self-restraint is as crucial as the capacity to attack, ensuring that the cure does not become worse than the disease. Your immune cells can live, migrate, and work in near-total darkness inside your body. They navigate by chemical signals rather than light, sensing gradients and textures through molecular receptors, crawling through dense tissue without vision or sound to guide them. The interior of your body is a lightless world, and yet immune cells move through it with purpose and precision, finding their targets in the dark through an ancient language of chemistry and touch. Plasmacytoid dendritic cells produce massive amounts of interferon during viral infections. These specialized cells are the body's interferon factories, capable of secreting more of this antiviral protein than any other cell type. When they detect viral nucleic acids, they activate rapidly, flooding the surrounding tissue with interferon that warns nearby cells and activates antiviral defenses. They are early responders to viral invasion, buying time through chemical alarm. The immune system creates specialized responses for extracellular versus intracellular pathogens. Bacteria living outside cells are targeted with antibodies and complement, while viruses hiding inside cells require cytotoxic T cells to destroy the infected cell itself. The location of the pathogen determines the strategy employed, and the immune system reads these contextual clues to deploy the appropriate arsenal. Different enemies require different tactics. Your gut-associated lymphoid tissue contains more antibody-producing cells than the rest of your body combined. The intestinal immune system is the largest component of the entire immune apparatus, a reflection of the constant microbial exposure the gut endures. More plasma cells secrete antibodies here than in all lymph nodes and spleen together, creating a dense antibody barrier that coats the intestinal surface. The gut is the front line, the border, where most encounters with the external world occur. Immune cells can recognize stressed or damaged cells by detecting unusual surface proteins. Cells under stress express proteins that are normally kept hidden, And these stress signals alert immune surveillance that something is wrong even before infection or transformation is complete. This early detection system allows the immune system to intervene before damage becomes irreparable, eliminating troubled cells before they become dangerous. The immune system has evolved over millions of years, refining its strategies against ever-changing threats. What protects you today is the product of an unbroken chain of survival stretching back through countless generations. Each ancestor's immune system tested by the diseases of their time. Those who survived passed their defenses forward, generation after generation, creating the sophisticated system you now carry. You are the inheritor of an ancient legacy, shaped by struggles you will never know against enemies long extinct. Your immune system works in silence, in darkness, and without rest. An invisible guardian that never sleeps. While you move through your days, while you rest in your nights, while you dream and think and live, billions of cells continue their patrol. They ask nothing of you, require no conscious direction, offer no complaint. They simply work, ceaselessly and silently, protecting the organism they constitute. You are never alone, never undefended, never without this tireless watch that began before your first breath and will continue until your last. And so our journey through the hidden world within draws to a quiet close. We've drifted through rivers of lymph and blood, past billions of silent guardians working in the dark, through memory halls where decades of battles are preserved in molecular form. We've witnessed the slow, patient dance of cells that know enemy from friend, destruction from repair, self from other. It's humbling to realize that while you rest here now, while your mind begins to drift towards sleep, this vast invisible universe continues its work. Your immune system asks nothing of you, no conscious thought, no effort, no attention. It simply protects, repairs, and remembers. Every breath you take, every moment of peace you feel, exists because trillions of cells are quietly devoted to your survival. You are never alone. You are a universe unto yourself, teeming with ancient wisdom and tireless protection. If you've enjoyed it, you can gently like, subscribe, or leave a thought in the comments. But for now, there is nothing more to do. Just allow your body to sink deeper into rest. Let the day fall away behind you, and let the calm widen within you, like the slow flow of lymph through peaceful tissue. Your guardians will continue their watch while you dream. There is no need to think, no need to remember, no need to hold on to anything at all. Sleep well, protected and whole. Good night.